All right, everyone. Thank you for coming and checking out the podcast again. Today's guest is Amit Deshwar, and we're going to talk about RNA therapeutics today. When I think about topics that are talked about quite a bit in the media and in the headlines and even just like around the dinner table, but that are simultaneously entirely misunderstood and no one actually contributing to those conversations has any idea what they're talking about, RNA therapeutics might be the most the topic that most accurately is described in that way, because because of COVID and everything that happened in the last few years, there's been a lot of sensationalism, both positive and negative, about mRNA therapeutics. And just yesterday, there was more sensationalism with the Project Stargate announcement and Larry Ellison talking about how we're going to make these like RNA cancer vaccines and all this stuff. So this is a topic that you can't avoid, and I think it makes sense to be educated on that topic. And Amit here is the founder of Kerna Labs, which is a startup that, let me see if I can remember this correctly. You are developing AI foundation models that enable RNA therapeutics to be used, or RNA to be used as the Universal Toolkit for genetic medicine. Is that the tagline more or less?
Yes, that's right. That's it.
Amazing. And you guys just kind of came out of stealth with some financing from Gradient Ventures and also the fund that I run as well, which is in the process of being renamed. So you're probably the expert that knows more about this than anyone else that I know, and so I'm excited to talk to you, man. Thank you so much for coming.
Excited to talk about RNA, which is something that I've spent a lot of time working on.
Yeah. So let's start there. We're going to get into all of the questions like what even is RNA, what is an RNA therapeutic, how is it different than normal drugs, but starting with the backstory, how does one become a guy that is leading a startup developing AI foundation models for RNA therapeutics? What's the backstory for you?
Well,
I
started my career as a software engineer. I was working at Google. I was building power supply firmware and log parsing frameworks, and I realized that I wanted to learn a lot more about machine learning, how to build these methods, how they work. And so I went to grad school. I did my PhD at the University of Toronto, just as the deep learning revolution was starting to happen. And when I started my PhD, I had a choice of, okay, I knew I wanted to do machine learning, but where did I want to apply it? And for me, applying it to problems in biology and medicine seemed like the right thing to do. And it was a combination of the fact that the impact that understanding health and biology can have is just huge and deeply meaningful to me. That if you look at all the increases in human well-being and health from the past 100 years, it's almost all been driven by our ability to understand and control biology, whether it's vaccines, it's understanding plant genetics. All those things have led to huge impacts. And then the second part is like a marginal contribution argument that people have been trying to build machine learning methods for language and vision for a long time. These are people who are very smart. They understand a lot of things. And they've been trying since the 60s to understand it. And in contrast with biology, it's still, I would say, vastly underinvested in, both in industry and in academia, that if you go to a big machine learning conference, the proportion of work focused on biology is maybe 1%, 2%. It's really small, which I think is vastly disproportionate to the potential impact that it can have. And so because it's so underinvested in, the chances that my work was going to be impactful, I think was quite a bit higher. And then coupled with that is that the data is brand new, that every year, coupled with the huge fall in sequencing costs, we have new ways of measuring and generating data about biological systems. And so a lot of this data, literally no one has looked at because it didn't exist until 12 months ago. And so those two things together meant that the impact that my work was going to have seemed like it was likely to be higher than working in vision or text.
Fair enough. And why do you think that only 1% of the talks at machine learning conferences are about bio-related subjects? Is it because one has to kind of be an expert in both bio and machine learning, which is quite more difficult than just being an expert in one in order to do that kind of research? Is it because, it's very hard to gather data for these types of problems? Is it because that there's a lot of bureaucracy that gets in the way of like making this kind of thing happen? Like what do you attribute that underinvestment to?
I think it's the combination of both business model and just data availability that combined together makes it harder for people to make progress in. That if you build like a chat bot, or you build a system that analyzes text to detect credit card fraud or something like that, there's really a direct and not easy but kind of straightforward path to commercialization. Correct. that depends like exactly on how good your model is. With biology, even if you build like the world's best model and it's finding these amazing drug like molecules, the path from like finding those to getting it into people and improving their health is separated by years and hundreds of millions of dollars. Right. In a way that other application areas aren't. So that's like kind of the challenges of the business. And then there just isn't the same large high quality prepackaged data sets in biology like there is in language and vision. And like if you think about how impactful ImageNet was, it was because they just built this huge high quality data set that people could use easily, that was recognized as an important benchmark. And then suddenly all this progress happened because that data was there.
Yeah. And for those who are not deep down the machine learning rabbit hole, ImageNet was basically this giant collection of images that were scraped from the internet that were labeled. So it was like if there was a picture of a dog, it was accompanied by a text that said dog. And so people could use that data set to train models. And basically there was nothing like that for RNA. And obviously it's much harder to get large quantities of biological data than it is to just scrape the internet for pictures of stuff. Yeah, that makes sense. But you were willing to brave these headwinds anyway, so now here you are. And so you worked at Deep Genomics for a while, and then at a certain point you decided to start your own company. What was sort of the insight? And we're going to get into like all the basic RNA stuff in a sec, but what was sort of the basic insight or moment of inspiration where you were like, now is the time for me to start doing my own thing? Was it something you saw in the market? Was it something you saw in the research world? What made you kind of take the plunge?
Well, when I joined Deep Genomics as one of the first employees, and my intention always was that I was going to found at some point. I just felt that coming out of grad school, I didn't really understand enough about how business works, how startups work, how to manage people, how to run teams, how to operate at bigger scales, and so wanted to get experience with that first. I spent almost 8 years at Deep Genomics, mostly building machine learning models of RNA, figuring out how to use those to find targets and find drugs. And we accomplished a lot. We built kind of the first large-scale RNA foundation model called Big RNA, which we put as a preprint out in 2023. But I was always in the back of my mind was surveying kind of all the 2010s AI for drug discovery companies and seeing them not have the impact that they intended to have. And it's not that I don't think they will succeed. I think multiple of these companies will develop successful drugs that will improve people's health. But if you add up, how much capital was spent, how long did it take, and is that, an order of magnitude better than the rest of the industry, which is what everyone intended to do when they started these companies. I don't think we've seen that yet.
I don't know if this is on Twitter or something, maybe you can verify or unverify this, but someone said something to the effect of, despite there being, probably hundreds of AI for drug discovery companies founded over the last 15 years, the number of FDA approved drugs that were discovered by AI is either 0 or 1.
I mean, I think the definition of what counts as AI discovered is like highly controversial. I've seen probably a dozen press releases being like, this is the first AI discovered drug in the clinic. And yeah, what does it mean to be AI discovered? I think can mean different things to different people. There are multiple drugs that I think you can plausibly say were discovered only through AI that are in the clinic and that appear promising. And so I don't think that that zero of these will end up working. I think for me, what was the big point of irritation was, is this actually way better than everything else? And I'm pretty confident that so far it hasn't appeared that way. That if you add up, all the time, all the research, all the money spent, that the productivity might be better, which is good, but the order of magnitude increase in productivity, which people were hoping for, has not yet appeared.
In fact, it's been the opposite, right, with Aram's law.
Yeah, so Aram's law is a really important concept where, in most fields, especially, adjacent to computation, and even adjacent to biology, there's this rapid increase in productivity over time, or, fall in sequencing costs, fall in compute costs. And for drug discovery, it's the opposite, where we are continually worse when you measure it as dollars in R&D per approved drug. It's been falling so the dollars have been going up continuously since the 60s, and at this point we're about 100 times less productive at developing drugs than we were then. And that's a problem because, it's really, at the point where it's like barely generating like positive returns. And if you have this vision that we're going to improve people's health, like that needs to generate positive returns. And so, the productivity has been getting quite a bit worse. Some of it is okay. There's more regulatory costs. You have to do more testing compared to what people were doing in the 60s. But that doesn't explain all of it, and it doesn't explain just how low the probability of success is. That if you look across the industry, it's between 5 and 10% of drug candidates end up getting approved, and the biggest lever that we have to increase that productivity is to change that probability of success. That yes, it'll be useful if you can run a clinical trial for less money. But the single most important thing we can do is change the probability of success. And there's one company in particular that was and still is very inspirational to me. They're called Alnylam. They develop also RNA drugs of a different class called siRNAs. And what is remarkable about Alnylam is that they actually have a high probability of success. So they've had 14 successful phase 3 trials in a row. They have each phase each clinical trial phase transition, their probability of success is above 80%, 85, 90. And their overall probability of success is about 65%. So it's 10 times better than the industry as a whole. Some of it is the diseases they go after. So far, all their approvals have been in genetic disease, which is simpler in some aspects. But a large part of it is that they've actually succeeded in building a reusable platform for drug discovery. And seeing that, watching it go from one approval, I think their first approval was in 2020 or 2019, and then watching that continue to succeed, like one approval per year since then, was like, oh, it is possible.
Yeah.
that it is possible through a combination of picking the right diseases, picking the right modality to have a high probability of success. And that was what inspired my decision to start Kerna, that I think the same thing is possible with mRNAs, and that coupled to that the way to make that possible was to use these machine learning models to understand how RNA works.
Yeah. Which are now just kind of starting to become widely available and understood. So
yeah, and that was the other thing, was seeing this huge step change in the ability to understand RNA biology by using transformers and the new architectures coupled with the scaling of compute availability and our ability to generate data, kind of all those things were happening at the same time. And so it seemed like the right place to be working in.
That's awesome, man. And so you've been on the journey for about a year and a half now, and we'll talk more about the company later. But let's get to sort of just like the background knowledge necessary to understand this. I think a lot of people, kind of know that RNA is vaguely related to DNA if they sort of like think back to that class in college that they barely paid attention to. But let's start there. The most basic question, like what is RNA? Why is it important to human biology and health? And then we'll talk about RNA therapeutics.
So RNA is a critical intermediary step between the information in your genes, in your DNA, and then the protein that DNA ends up producing. This is, I guess, a repeat of intro biology where you have, your gene, your DNA, a copy of it gets made that is an RNA molecule that's a direct copy of what is there in your DNA. It gets modified in a few ways. Chunks get cut out, pieces get added to the end and the beginning that make it ready to be turned into protein. And then your ribosome, which constructs proteins, reads the information contained in the RNA that tells it, to add a series of amino acids that make up a protein. RNAs have many other functions. There's a whole array of RNA species, but the RNAs that encode for proteins are most understood and kind of most central to our biology.
So would a correct analogy sort of be like the DNA is maybe the entire code base of like a software program or a website or something, and then maybe the RNA is like a specific like API query or like message that is, or like way of interacting with that program or like subset of instruction, and if that's not the right analogy, what is like a good analogy that someone could use to try and just quickly grasp what's happening in a more or less correct way?
Maybe the way to think about it is if the DNA is the source code and the protein is the executable, the RNA is like a combination of like symbol tables and parse trees that lie in between the source code and the executable.
Got it. So it's sort of like a translation step between the source code and the actual thing that happens from that source code. That makes sense. All right, so that's intro to RNA. And then RNA therapeutic. What exactly is an RNA therapeutic? I think when people hear that term, they might think, are we using RNA as a therapeutic? Are we using some other type of drug to like fix broken RNA? Maybe let's start there at a broad level. Like what exactly is an RNA therapeutic?
Yeah, I mean, RNA therapeutics, I guess I would say, are therapeutics that are composed of RNA molecules that we make in the lab or purely synthetically, and that when introduced, interact with the rest of our biology that already is designed to work with RNA to have a therapeutic outcome. And there's a few like major classes. There's siRNAs, which I previously mentioned.
That's the one that company, Ellison, is using as their modality. And you guys are doing mRNA, which is messenger RNA. Okay.
Yes, yeah. There's also oligonucleotides developed by companies like Ionis and Biogen. They all have this common thing that the chemical composition is RNA, and we can make these RNA pieces with a sequence that we control, and then once they're inside your cell, produce some biomolecular effect.
That makes sense. And so, going back to sort of just like how the average person, me included here, understands like biology and drugs. Like someone who's not trained in the life sciences, I think most people vaguely understand drugs as like, there's a molecule that looks sort of like that thing that was on the cover of the chemistry textbook that they barely paid attention to. And maybe that molecule is caffeine, maybe it's aspirin, maybe it's THC, maybe it's something else. But you ingest it through a pill, or maybe you drink it in some liquid form, and then it kind of like floats around in your bloodstream and it binds to receptors, which I think most people are kind of vaguely familiar with because people use terms like, this drug like boosts my serotonin or dopamine or something by binding to these receptors. So that's like what most people think drugs are, and most drugs are that. But RNA therapeutics or mRNA therapeutics, very, very different. And we'll talk about maybe some of those differences, but maybe a good place to start is like, why bother making mRNA therapeutics? Like, why can't we just use these traditional small molecule drugs to treat all the diseases? Like, what are we able to achieve by therapeuticizing mRNAs?
So what you can do with mRNA therapeutics is you can produce a protein that you want. That's like the base ability of mRNAs. That rather than relying on your body to transcribe your DNA and then turn that RNA into a protein, we can introduce an RNA that encodes for a protein that we choose. And that enables you to, everything inside our cells operates on the level of protein, like the receptors that we talked about are proteins and the molecules bind to them. But with this, we can introduce a protein that we want. And so that's what enables like mRNA vaccines, which many people have taken, where you encode for a portion of the COVID protein. That mRNA goes into your cells. Your body produces this protein that is very similar to what the COVID virus would do. And then your body recognizes that there's a foreign protein, builds up an immune defense for it. And then when you encounter a real COVID virus and protein, your immune system's already ready for it. And you can't produce an arbitrary protein by adding a small molecule. You can bind to receptors, you can change levels of your natural transcripts, but you can't produce arbitrary proteins using small molecules. And that's the power of mRNA.
So yeah, I want to ask the question of like why. Let's start there. So like, why is that sort of why the COVID vaccine had to be an mRNA vaccine? Like there really weren't any other choices, or was it just that doing it via mRNA vaccine was far easier than any of the other possible avenues?
I mean, there are other approaches to doing vaccines other than mRNAs. Like we had vaccines before. You can encode the protein in a DNA, and so like the Janssen COVID vaccine, I think was a DNA vaccine. Very few people ended up taking that, and I'm pretty sure it was pulled from the market. A combination of safety and effectiveness compared to the mRNAs just wasn't the same. We can also do things like make less infectious versions of the virus and have people exposed to that. So there's other ways of doing of producing proteins, but in terms of the flexibility, the safety, and the general purposeness of it, mRNAs can just do more than most things.
I want to throw this analogy out there just because a lot of the people listening have listened to some of the episodes I've done on psychedelic drugs. So would it be fair to say that like a psychedelic molecule, it might bind to a specific serotonin receptor, which is on the membrane of your cell somewhere. But an mRNA therapeutic, in theory, you could make one that would cause your body to create even more serotonin receptors. So that way, serotonin could cause more activity. Is that kind of the correct way to think about it?
Yeah, I mean, but
you're
it's
not
more
binding to the receptor, but you could, in theory, make more or less of those receptors.
So you can make more of a specific receptor, but you could also make a variant of that receptor. So maybe you produce a serotonin receptor that is much easier to activate, or is just activated all the time. And so there's many more things you can do, even in that realm.
And so outside of the COVID vaccine, actually this is like a bit of trivia that I have no idea what the answer is. Was the COVID vaccine like the first mRNA medicine? Or were there others?
I'm pretty sure it was the first. Well, no. So Alnylam had siRNAs that were approved before.
Right.
In terms of mRNAs, I'm pretty sure it was the first approved therapy. Was the COVID vaccine? People have tried to use mRNAs for other things, and there's some promising data of using mRNAs to deliver things like CRISPR editors, where we want to produce this novel protein to edit your genome. And the best way so far to do that is to encode that editor in an mRNA and deliver it that way.
Interesting. And so, I mean, outside of like COVID, would you kind of explain why it was easiest to make an mRNA COVID vaccine versus other avenues? What are like specific disease categories that particularly lend themselves to be treated with an mRNA therapeutic versus the classical type drugs that we are more familiar with?
I mean, the main advantage of genetic medicines is the specificity of the intervention that you're able to do. That, if you have a small molecule binding to a receptor, it's very hard to get it to only do one thing. Like if you look at drugs like psychedelics and even anything that's affect your cognition anyway, it's typically binding to many things. Right. And with the mRNA, you're only producing like exactly the protein that you want.
And when binding to many things is a nice way of saying that's where the side effects come from, right?
Some of it's side effects. I mean,
right?
Sometimes you actually want like polypharmacology
Sure.
where it's helpful to be binding to multiple things, but it's hard to design that in the exact way that you want. Like you end up with the binding profile and you're trying to like make it bind more or less to different receptors, and it's really hard to do. And with mRNAs, you're like, okay, I want to produce exactly this protein, and that's exactly what you get.
And so when you think about going back to like this compare and contrast between traditional drugs, which are small molecules, and RNA therapeutics, I think people that at least have paid attention in chemistry, it's like you've got the drug, maybe you have this idea of this molecule because you took it out of a plant that people kind of knew this plant had this effect. So like the classic example being aspirin, which was from the willow bark tree. So they like isolate it. They're like, okay, cool. Here's like this chemical structure. It's simple enough that I can draw it on a on a chalkboard. And because of the knowledge of chemistry that humans have built up, we kind of know like, oh, maybe we like mess with this little like ring system over here. Maybe we like replace this hydrogen with a deuterium, whatever. These little like substitutions or modifications that can be, and for many years were done by hand, no computer whatsoever involved. And it was like a small enough problem that it could like fit in the brain of one or two smart chemists, right? With mRNA therapeutics, like how does one even begin to go about designing the mRNA sequence itself? Because my understanding is, and correct me if I'm wrong, the size of the universe or the number of theoretically possible RNA sequences is probably a lot bigger than that of the possible small molecules. Is that fair to say?
It is definitely bigger, but I'll say that the search space of small molecules is also quite large.
It is big.
Like I think people say like 10 to the 60. I'm not a medicinal chemist, so I wouldn't count on that too much. But it is still quite big, and we were able to take advantage of evolution and what we were able to see from natural biomolecules to understand some of these rules, to have good starting points for molecules that were going to be active. Yeah. And we, for mRNAs, like historically, we've done basically a very similar thing where the RNA sequences that are already in our genes or in viruses are a good starting point. And so for the COVID vaccines, that's what they did. They took the protein that they wanted to encode, and then they surrounded it with sequence that is from a human or derived from a human gene that's naturally highly expressed. And they were able to, come up with the sequence in 2 days or something like that. Once COVID was sequenced, it's quite a remarkable story. So for many applications like vaccines, we can do that. We can take what's already known, combine it with the protein we want to produce. We know some rules about how to encode that protein, and then you have an mRNA.
Got it. So it is sort of possible to do some like by hand RNA therapeutic development.
Yeah, and that's why we have mRNAs.
Right. Okay.
and the reason, to go back to, okay, why am I building these models in the first place, is that outside of vaccines, and then looks like the CRISPR programs that I mentioned, mRNAs have not been successful historically. That people have been trying to use them for other diseases like genetic diseases. And so far, we don't have anything that's approved and is clearly benefiting patients. And the problem is that with the vaccines, in some ways, I don't want to trivialize like all the work that people did to create the mRNA vaccines, but they are mRNA therapeutics on like easy mode, where you just need to produce some protein for a short amount of time in any cells. And then your immune system is so sensitive and capable that that's enough to have an effect. For other cases where you're not able to leverage the immune system in that way, you need to produce higher quantities of protein. You need the mRNA to last for longer, and you need to produce it in the cells that where you want it to be. And that's where we haven't been able to find the right sequences so far. And that's what Kerna is trying to solve. That out of the huge search space of possible mRNAs that encode for a protein, we want to find the ones that produce enough protein, that last for long enough, and that produce in the right cells so they can be effective therapeutics.
And you're building these foundational AI models to enable that discovery. I mean, maybe you could talk a little bit about that, the tools that you're using, these foundation models, and you could sort of use the analogy of ChatGPT and language models, which I know are probably very similar, but not identical to how you're approaching the problem at Kerna. Do you want to just kind of go from there?
Sure. I mean, by analogy, it's really a very similar process where we are also using transformers and related architectures for the models that we're training. But rather than training them on text and to predict text, we train them on RNA sequences and try to predict RNA properties from it.
I see. And so you're not just like in the ChatGPT world, you're basically predicting the next word. So the quick brown fox, and ChatGPT learns jumps and then over and then the, right? But you're not necessarily doing that sequence by sequence. You're like base pair by base pair. I don't know if that's the right term, but your sequence is the input, output is different properties of the mRNA.
Yeah, I mean, I would say that so with the language models, they have to do a multi-step training process where they start by just trying to predict the next word, and that's what gives them their base model. But the base models are not that useful directly. To go from GPT-3 or GPT-4 to ChatGPT, they had to do what's called RLHF, reinforcement learning from human feedback, to tune the model to respond not just for like what is the most likely next word, but what is the most likely next word in a chat conversation. And so we do something similar where we train this big base model on kind of all the RNA data that we can get our hands on. But all that data is natural RNA. Like, the same.
It exists in nature.
It exists in nature. It exists in our bodies already. The mRNAs that we use as therapeutics are different chemically in a couple different ways. They have different caps, which is on the start of the transcript that tells your cells this is something to produce protein from. We don't use the exact same bases that natural RNA uses. Rather than uracil, we use pseudouridine to encode for the U base.
Is that just because it's easier to like synthesize that or manufacture it, or what's
the use of pseudouridine has two important effects. One is that it stops the RNA that you introduce from being detected as a foreign RNA. So your viruses are RNA. Many viruses are RNA viruses. So if your immune system detects some foreign RNA, that triggers an immune reaction, and then your body will try to degrade the transcript, and that's not what you want if you're trying to develop mRNA therapeutics. So the pseudo-U prevents the immune system from recognizing it as foreign RNA, and it also increases the amount of protein being produced by that mRNA compared to natural sequences.
And I assume this is not something that's specific to Kerna. This is like a known thing.
This is so like the COVID vaccines that you took, unless you got, there's one strange one that's natural, but the Moderna and the Pfizer BioNTech vaccines were pseudo-U modified.
I took the Sputnik Russian one. I don't know what that
okay, I'm not sure what that one is either. I think it's CureVac. I think they have a natural RNA COVID vaccine that was, quite a bit less successful than the pseudo-U ones.
So that's one of the differences. You use that compound versus the naturally occurring one.
Right. And so that's why we have to do the second step in the training process to fine tune on mRNAs that are chemically similar to the ones that we want to use. So there's the base model, and then there's the fine tuning on the chemically modified and other differences to the mRNA. Just like with the chat models where they train the base language model and then do RLHF to turn it into a chatbot.
Yeah, and just to like make the analogy more clear, so like with ChatGPT, the RLHF step that you're talking about, the reinforcement learning from human feedback, was literally they had thousands of people, sitting in a room, basically using ChatGPT, and ChatGPT would they would interact with it, and they would basically press this is a good response, this is a bad response, and the model would get a little bit better that way. What is the RNA version of that, or the mRNA version of that?
So we synthesize mRNA molecules and measure their properties, which are what I was talking about earlier: half-life, protein output, tissue specificity, and then give that information to the model to learn the specific rules for the mRNAs that we want to use.
And so is sort of like the if you think about with going back to the ChatGPT analogy, like the input is, maybe a question that's typed via text and the output is an answer. Is it fair to say that in the Kerna case, or just in general, if one was to create a model to do this, is it like the input or the question that's being asked is the protein that you want to express, and the output is an RNA sequence that will tell the body to create that protein? Or
it's that's the generation that we want to do, where we have an amino acid sequence we want expressed, and then we have properties that we want the underlying mRNA sequence to have, and then the model's trying to generate that. And so the training data is mRNA sequences and the measured properties. Right. And then so we use that to teach the model, okay, given this amino acid sequence, given the mRNA sequence, here's the resulting properties. And so learn the rules that are underlying it.
Yeah, learn the rules, and then you can probably use that to create a number of candidates, and then you pick the ones with the best properties, and then maybe there's a little bit of human fine-tuning at the end there or something like that.
I mean, there is definitely, an experimental part.
Sure.
It's not to the level where we can just give it the amino acid sequence, it picks one sequence, and then we're ready to go into the clinic.
Yeah,
we're not at that stage yet. You still have to measure it. You still have to test it. And we can, as we're developing our own therapeutic programs, we use all that data that we're generating to make the model better. Sure. And so for the next programs, we'll be able to be even more effective at finding the right sequences.
And so what do you think, not necessarily asking about Kerna specifically, but what diseases are you most excited to be able to potentially cure with mRNAs?
I mean, in principle, mRNAs can treat almost everything. And that's really the power of genetic medicines. And so there's, different types of diseases with different, underlying biological complexity. The most straightforward application is in genetic diseases, where the disease is caused because you are missing a specific protein. And so we can encode that protein in the mRNA, give it to you, now you have that protein.
So what, just for the non-MDs out there, what is an example of a genetic disease that right now you can't really cure, but in theory you could with an mRNA?
Sure. I mean, hemophilia is maybe the most well-known genetic disease. And also, we have some effective therapies for it, so I don't think it's not that it's untreated today. But that's, I think, the most well-known genetic disease. So hemophilia is a bleeding disorder. It's caused where you are missing a protein that's necessary for the clotting response. And so when you get a cut, clots don't form.
You just don't stop bleeding, basically.
Yeah.
Quite dangerous.
Yeah. And so that's something that you could treat with an mRNA. But what I'm most excited for is moving genetic medicines from beyond genetic disease to the rest of the diseases out there. That genetic diseases are very important, they're very severe, but the impact on overall population well-being and health is going to be driven by solving non-genetic diseases. I see. If you look at the impact of GLP-1s, which of course many people are aware of now and might be taking, that the aggregate impact on human health by through GLP-1s is just way bigger than really anything else, and it's because the most common diseases tend to be not genetic, and so for Kerna, that's really where we're putting most of our emphasis on is how can we take a genetic medicine approach and use it to solve big diseases that are going to have a big impact on human health.
Without giving away any of the secret plans, can you name any specific, at least maybe disease categories that are not genetic diseases, but that you think might be more easily treated by mRNAs? Or what lends itself most naturally?
If you just look at what are the major killers, and metabolic disease is of course major killer, neurological disease, major killer. There's many out there, and all of them are affected by our genes. That if you look at any disease, any disease, including genetic and non-genetic, the mutations and the differences in the DNA that you have change your ability to either resist or to get that disease. And so that indicates that by using a genetic medicine, you can treat or prevent a wide range of diseases.
That makes sense. Yeah, and I guess sort of going back to that, not really analogy, but example that I was talking about with what was I going to say? With the psychedelics and how one could theoretically, increase the number of serotonin receptors using an mRNA drug, or maybe, like you said, design a new sort of type of serotonin receptor. This is an example of how a genetic medicine could potentially be a treatment for depression, as opposed to an SSRI or something. Is that fair?
Yeah.
No,
exactly.
Okay. That kind of brings it back. It's very interesting, man. And so what are obviously this all sounds amazing. What are some of the constraints or challenges? I know, for example, in the small molecule world, if you want to treat a disease in the brain, there's this issue with the blood-brain barrier, and a lot of molecules are too big to kind of cross the blood-brain barrier. So even if you design the perfect drug, you can't really get it into the brain. And I'm, some drugs, they just are not stable. And so by the time you even get them in the human, they've already degraded. What are some of the challenges with mRNA therapeutics that are in that similar kind of vein?
So beyond the problems I was talking about earlier of protein output and half-life and cell type specificity, delivery is also a major problem. Mm-hmm. That mRNA molecules are quite large, much bigger than small molecules. They don't, passively diffuse across cell membranes in the same way that small molecules can. And so getting your mRNA inside the cells that you want to produce the protein in remains a major challenge.
I see. Because it has to get inside the actual cell in order for it to do anything.
Right, because that's where the ribosomes are. They're not floating. They're not floating around. They're inside your cells, and so if you want protein to be produced, the mRNA has to get inside the cells.
Yeah. And just for people who are not super deep down this rabbit hole, a lot of the receptors that small molecules bind to, they're sitting on the cell membrane. So you don't have to get into the cell. They're just kind of sitting there on the outside of the cell. So how do they get in the cells? What's the state of the art in terms of?
So the most common way is you package the mRNA inside what's called a lipid nanoparticle. So it's a small bubble of lipids that contain the mRNA that binds to either receptors or the cell membrane itself, and then the mRNA goes inside the cell. And innovation in LNPs is what has made mRNAs possible in the first place. And that remains this huge area of research. Both, can we come up with new LNPs that will reach more cells? Can we add things to the surface of the LNP so that way it goes to the cells that we're interested in?
Is that what the COVID vaccine was wrapped in? Or was
yes. So the COVID vaccine the mRNA was wrapped in a LNP.
I see. Is it fair to assume that most genetic medicines, including mRNA therapeutics, are going to be vaccine delivered, or could you, in theory, have oral, tablets or nasal sprays or something?
Oral is tough. Your digestive system is very good at chewing things up, which will make the mRNA not effective. It's chewed up into its constitutive parts. inhalation is a very interesting potential way of delivering mRNAs. That your lungs have big surface area, and you can potentially introduce LNPs, wrapping around your mRNA through inhalation. That is simpler and easier to do than a intravenous injection, or even the intramuscular / sub-Q that we did with the vaccines.
Interesting. So if you're at a party and you see a guy wiping his nose, maybe he was just doing some mRNA.
Could be. I mean,
okay.
not yet. Not yet.
Someday.
It would look like the people in like the oxygen therapy where you're breathing it in. Yeah. We're not there yet, but it's coming, and there are multiple companies either close to the clinic or in the clinic with inhaled mRNAs. So that's coming.
Makes sense. Well, I feel like we're kind of getting towards the end here. I do have a few other things I wanted to ask you about. I don't like talking about the recent news so much because that often makes the podcast very dated very quickly. But we are just one day after, this Project Stargate announcement happened, and there's a clip of Larry Ellison. I think it was him, or maybe it was Sam, but basically just like, we're going to be able to create these vaccines for everything, including cancer, and it's going to be personalized, and we'll be able to just, swab your DNA, take a look at what's going on in your cells, and within 24 hours, you'll have like a personalized vaccine that's like ready to cure whatever ails you, basically, all through the power of AI combined with genetic medicine. What do you think? I mean, is this way too hyped? Is this underhyped? Like, what is your sort of take on this whole Project Stargate announcement and the vision that Larry and Sam are throwing out there?
Well, I think the funny thing about that clip and the statement is that that's already here. that he's talking about as something in the future that only AI will enable once they spend $500 billion on data centers, but Moderna and BioNTech are already in the clinic with personalized cancer vaccines. So for each patient, they take a sample of the DNA, they sequence it, they figure out what new antigens are presenting in the tumors versus in your healthy cells. They make a custom mRNA vaccine mixture for your cancer. And it's still early. I think they finished phase 2s maybe. Seems to be effective. So that's already here.
So
he's like 5 years. He's 5 years. 5 years in the past.
But you're actually saying, no, dude, we're already doing this. What are you talking about?
We're already doing it. Already in the clinic with personalized cancer vaccines. And so, if anything, he's like, 5 years behind what's actually happening.
So let me ask a slight derivative of this question. It is the lack of $500 billion in AI infrastructure holding us back, or are the limitations of genetic medicines becoming more widely available and progressing further in the clinic? Is it due to non-AI infrastructure reasons?
I think the bigger bottleneck for what we're doing right now is on the data side. So generating mRNA data of sufficient scale and quality to train even better models. Gotcha. And if you look at the state of the art of any model that's looking at biology, they're way smaller than the big language and vision models that companies like OpenAI and others are developing. And some of it you might say is like the investment and like, do they have enough money for the compute? But I think the real reason why companies like DeepMind haven't scaled their protein models to be orders of magnitude bigger is just how much data do they have to train on. And that's, I think, by far the biggest bottleneck is just enough, high quality, relevant biological data to train these models.
So do we need, like, rather than Project Stargate, do we need like maybe not even 500 billion, maybe just a few billion of government money to go around and collect a bunch of data? That would be better? Yeah,
I mean, I think that because data is the biggest constraint, that whatever we can do, whether it's private companies, whether it's big government projects to generate biological data, are key to making progress. Projects like the UK Biobank and GTEx and all of us are big government projects to collect DNA and RNA data from many people, and are basically like bedrocks of anyone trying to build these models. That having the DNA and the RNA sequenced from many individuals and available is key to building these. There's been a large large is relative now, but like say billions spent on sequencing so far, and I think that's going to continue to increase and is going to be key to understanding biology.
Amazing. Well, I'm going to short Oracle right when we're done with this podcast. But this has been really fun, man. There aren't any obvious questions that I feel like I need to ask at this point. Was there anything you wanted to talk about that we didn't get a chance to touch on?
Well, I think that the premise of rapid personalized drugs is something that is already starting to happen, but is so far just in the cancer vaccine world. And the reason why it's possible is because of the flexibility and kind of class general properties of nucleic acids. Mm-hmm. That it's not a huge mystery. If I have a new mRNA sequence, how am I going to make it? Where is it going to end up in the body? How high can I dose while it's safe? And because those are not exactly constant, but close enough to being constant, that's what makes personalized vaccines possible. And that's what's going to make personalized medicine as a whole quite possible. There's a very interesting story of a drug called Ellison, which was a custom oligonucleotide drug that a patient, there's a physician called Tim Yu, he's at Boston Children's. He met this patient, figured out what was the genetic cause of their disease, and then developed a custom oligonucleotide for that patient in about 10 months, from seeing the patient to having it given to her.
Wow.
Which is insane.
Yeah.
10 months.
And this is just a drug just for this person.
Just for that person.
Yeah, they have very rare genetic disease.
Exactly. So this was the first person to be seen with that specific mutation. And basically, I think that's possible for many, many more things. That the flexibility, the class general properties of nucleic acids and genetic medicines as a whole make it possible. And I think we're going to see quite a bit more of that.
Yeah, and it goes way beyond just here's someone with a devastating, probably terminal, extreme genetic mutation. It could be just that someone has a mutation that's relatively unique to them and doesn't cause, massive problems with their quality of life, but is causing maybe a small problem. And if you tweak it, then you can increase that person's vitality, energy, just like overall well-being. And maybe that's how you end up getting to this like longevity takeoff as well. Maybe not, but
hopefully,
we're in San Francisco and we met at
yeah.
the Foresight Institute, so we have to say longevity at least once on the podcast.
Well, I think if you look at even the most successful drugs, they have a range of outcomes that even the GLP-1s, many people can't tolerate them. For many people, they don't have a strong enough effect, and these are some of the most successful drugs. And so the reason why is because we're not all identical. And so developing medicines for our specific biology is how you're going to fix that. So that way they work for everyone. Amazing.
Well, where can people find you guys online if they want to learn more, maybe potentially work for you or potentially invest in your company?
It's KernaLabs.ai. You can find us on Twitter, find us on LinkedIn. Reach out.
Awesome. Are you guys hiring at the moment?
We are hiring, particularly if you're a ML engineer or ML researcher that wants to build foundation models of RNA. We're hiring for that right now. So reach out.
Awesome. Thanks so much, man.
Thanks, Brian.